Hello everyone, my name is SilvinaCainoLores and I'm here on behalf of a team of researchers at the University of Tennessee, Knoxville, and several collaborators working on this project towards in-situ enhanced adaptive sampling of simulation ensembles through analytics from molecular dynamics. I would like to first acknowledge our funding agencies and sponsors, IBM and the National Science Foundation, and our collaborators from Cornell University, the University of New Mexico, and the University of Southern California also participating in this project. I would like to start first by giving a brief introduction to what a molecular dynamic system is and how we simulate them. So molecular systems are composed of atoms that forms structures in a hierarchical way. So typically one would have atoms forming a chain of amino acids that bind together to form more sophisticated structures like helices or sheets. And with these secondary structures, we build the molecules and proteins we are used to seeing. In order to understand what happens when these structures are exposed to external and internal forces, we conduct simulations that simply reposition these atoms at each point in time. Now, how do we understand what's happening in the evolution and life cycle of these proteins? Well, we take snapshots, we take pictures of what's happening in the system every given number of steps that we call stride. And we take this image of what's happening in the system, and I will refer to this as frames. When we put one frame after the other, essentially what we have is a discretized film that represents the evolution of the system through time. In this evolution, many things can happen, and there are particular events and structures that are very relevant for the domain scientists. There are transformations that we can see as shown in this first example, in which the atoms rearrange in particular shapes or conformations that have particular functional properties of high relevance, or simple movement in the across different structures of the molecule. All of them are representing relevant changes in the system. So typically what is done to understand and capture these events is to go through this state of these frames that I was mentioned before, and we capture what is called a collective variable, which is essentially a mathematical abstraction that reduces the dimensionality of the system. So instead of having billions of atoms, we reduce this to hundreds of collective variables that capture what is happening in the structure of the protein. And we compute those for each frame of this trajectory of this simulation. And by capturing that, scientists can visualize or can analyze what's happening in the simulation. Now, this is typically done after the simulation has completed. So one would have a simulation running for 24 or 48 hours, and then computing all these collective variables at once, and then trying to analyze what happened. What we try to do in this project is similar, but we try to remove the human from the loop. So we try to analyze the simulation as it is operating. So we have one component that is essentially the molecular dynamic code, and we are agnostic to the molecular dynamic code that we have. And as it is running, we will run our analysis that is capturing the collective variables per frame. And in the middle, we have in-memory staging area that is able to send information from the simulation all the way to the analysis. And this information is a frame in the simulation. So essentially what we do is, as the simulation runs, we're able to capture the frames and analyze them without disrupting the simulation. There is one additional benefit of this, And that is that we can, with this information, go back to the simulation and maybe decide to terminate it if it's not useful, or restart it with more promising parameters, or just store all the information that we are gathering and annotate the frames. So what I explained before works for one simulation, but typically in realistic scenarios, we have more than one, because molecular dynamic systems are chaotic in nature. Typically, we have hundreds of thousands of molecular dynamic jobs with the goal of capturing all the different conformations and transitions that a system can go through. So in order to support this, we have introduced many changes in our project to support not one but multiple simulations running concurrently with multiple analysis components. And this empowers us to conduct a higher level prediction and steering for the entire ensemble. So this has many applications. I'm going to start with one that is very useful for scientists, which is to understand and minimize the folding process for a given protein. In this particular case, we have a very small peptide. And we see here the conformational map. So this is a Markov state model for the protein. And as you can see, some errors are a bit thicker than others, meaning that there is a higher chance for the protein to jump from one state to the other. So thicker means higher probability. And what we know from this is that some simulations might not be very promising or may not have the probability to reach the folded state. So we worked with different collective variables to try to understand when this small probability is reached. So in order to simplify, you can see that for this collective variable, that is the largest eigenvalue of the distance matrix between the atoms, there is a particular range that indicates a lot of stability, meaning that the protein is not likely to jump to a different state. So if for a given window, in this particular case, 200 frames, these values remain in range, as you can see here, we can consider that this trajectory is one of these very low probability ones to reach the folded state, so we can terminate it. On the other hand, if this is not the case, we can continue running the simulation because it has a higher chance to transition. By applying this method, that we can analyze in situ, we can terminate early trajectories that are not likely to jump, which means that according to our experiments, roughly 50% of the trajectories can be terminated early, ranging from 20 to 60% of savings in molecular dynamics. That's with this and also other connected variables. Another example is, instead of minimizing the time to reach a particular form, is how much time do we need to cover the entire space of different forms that we can have. So in order to understand this example, we have here a use case for the human dopamine membrane transporter. This is a very large molecule, and this is a representation of the TICA space that captures the different conformations, the different shapes that this protein can have in its structural space. So we have developed methods to understand whether our trajectories, a simulation, is able to explore more or less likely this space. So here's an example of an unfavorable trajectory that kind of remains stuck in this area. And this is an example of a trajectory that jumps from one region to another. So these trajectories we can accept in our ensemble. Well, we can terminate the trajectories that are kind of constrained to a smaller region of space, which means that with a fraction of the trajectories, we can cover pretty much the entire conformational space, saving lots of computational resources. So just to summarize, I would like to remark that A-frame D is a tool that enables better knowledge discovery in the domain of molecular dynamics, because it allows us to inform our simulations and our models as the simulation is running. And this is also useful to make a more efficient use of the computational resources. We have a lot of work to do, including incorporating new relevant detection methods, also based on machine learning techniques, in terms of annotating, indexing, and supporting search of the intermediate and output annotation data, and also in terms of profiling, optimization, and enhancement of the orchestration of the different components of the workflow. So we would be really happy to talk with you about potential use cases and improvements to this tool. Thank you very much, and I hope I can answer all